Hello, my name is Helmut Messmann. I'm professor of gastroenterology at the clinic in Augsburg in Germany. I was asked by the editors of Gastroenterology to discuss a poster presented at the DDW in Chicago with the title Chromoendoscopy with Automatic Lesion Enhancement in Magnetically Guided Capsule Endoscopy, a Feasibility Study. The technique of chromoendoscopy is an established procedure, especially in Asia countries, but also in US and Europe. Chromoendoscopy has been proven to significantly enhance the visibility of mucosa irregularities like metaplasia or dysplasia, which are both precancerous lesions and can grow to gastric cancer, which is the second most digestive neoplasm in the world. Chromoendoscopy has been applied in a variety of clinical settings in different parts of the GI tract and involves the use of different stains and pigments. The second technique involved in this contribution is capsule endoscopy. It is also an established procedure for the examination of the small bowel and more recently for the colon and esophagus. Recent researches focus on steerable endoscopic capsule and their application in the stomach. Animal and human trials have been reported from Carter, Swain and Ray. In these magnetically guided approaches, the stomach is liquid distended and the patient swallows an endoscopic video capsule, which is navigated by an external magnetic field. The procedure is virtually non-invasive and comfortable for the patient and requires no sedation. Magnetically guided capsule endoscopy seems feasible and sufficiently accurate for the examination of the gastric mucosa. However, known difficulties in the diagnosis of metaplasia in the conventional endoscopic transfer to magnetically guided capsule endoscopy. Therefore, magnetically guided capsule endoscopy may also require the use of stains similar to chromoendoscopy. In this contribution, we aim to prove the feasibility of a staining procedure in a magnetically guided capsule endoscopy examination of the stomach, as well as the potential of automatic algorithms for further enhancement of the visibility of lesions and the mucosal in general.
Hi, my name is Mewes. I'm a doctoral candidate at the Pattern Recognition Lab of the University Erlangen-Nürnberg.
And I am Stefan Förtsch. I'm a doctoral candidate at the Department of Chemistry and Pharmacy of the University in Erlangen in Germany. Chromoendoscopy uses different staining procedures. We decided to apply methylene blue, which was a commonly used stain and was successfully used in various clinical procedures in gastroenterology. We used 100 ml of methylene blue with a concentration of 1 gram per liter. This solution turns out to perform best in staining of the stomach walls without degrading the visibility inside the water-filled pig stomach.
Our experimental setup consists in 3 steps. First, the pig stomach was filled with lukewarm water, as described in recent literature for magnetically guided capsule endoscopy. The pig stomach was prepared with lesion imitations of polyps and metaplasia. Reference images were captured inside the stomach using a given imaging SB2 capsule and a plastic tube support to navigate the capsule inside the stomach and to focus on capturing especially the prepared lesions, but also healthy mucosa. Afterwards, the stomach was emptied and the previously described staining procedure was applied. To complete the staining procedure, the stomach was filled with the same amount of water. Again, images were captured. As a last step, image enhancement method were applied to the images, taking into account the specific color spectrum achieved through the staining procedure. Afterwards, images were compared regarding the visibility of lesions and the general appearance of the stomach mucosa.
The results demonstrate that the proposed staining procedure can be applied to pig stomachs. Similar to the traditional chromoendoscopy, the mucosa appears more detailed. Image processing algorithms can further improve the image quality. Though the visibility of metaplasia and dysplasia is enhanced, other pathology may be masked. This, however, is acceptable for examination focused on the detection of mucosa irregularities.